Welcome to the MCAT Basics podcast, brought to you by the physicians at Med School Coach. Each week, Sam Smith breaks down high-yield MCAT topics so you can score as high as possible on test day.
Hello, I am Sam Smith, and this is Sam's MCAT Basics. This podcast covers the most important topics put out on the AAMC MCAT each year, and I determined this list by going through all the official practice materials that the AAMC puts out, and also some third-party practice material, and just put together this big list of topics that consistently kept on showing up, and ranked those topics and made podcasts out of it. This podcast covers biomolecule structure. I'm going to go over molecules like steroids, nucleotides, sugars, fatty acids, and I'm going to talk about their structure, and I'm going to talk about their function. And I'm going to get into quite a bit of detail. Things like naming the different phosphate groups on ATP, for example, or talking about fatty acids that act as signaling molecules. So there's some good stuff in here. I think it'll be useful for the MCAT. And this topic will show up on 2 out of the 4 sections of the MCAT, show up in the chemical and physical foundations of biological systems and the biological and biochemical foundations of living systems. In my experience, you will see multiple questions on both of these sections on the MCAT. So it's a very important topic, and I hope this podcast helps. All right, so I want to start by first saying this podcast is about the structure of different molecules. So I'm going to try to describe the structure to you, but at some point you will have to look up the structure, make sure you know what it looks like. And I think it will definitely help if you look up some of these structures before, during this podcast, and kind of make sure you know what they look like. That way you can make associations in your head. And it's just going to help you memorize different topics and understand different concepts. All right, so the first structure I want to talk about is the steroid or the steroid ring. So this class of molecules called steroids all share a common backbone structure. And the structure is 4 carbon rings or 4 hydrocarbon rings. And so 3 of these rings are 6 carbon rings, and 1 of these rings is a 5 carbon ring. And it's got a lot of hydrocarbons, very non-polar hydrophobic molecule, and that is where it derives a lot of its functionality from. So if you know anything about steroids, they act as hormones in the body, and they can diffuse straight across a membrane because they are so small and non-polar. And what they do is, how they act as hormones is they diffuse through the membrane and into the nucleus where they affect transcription. However, to be carried in the blood, these steroid hormones must be carried around by these little carrier proteins. And these proteins are called hormone-binding globulin. And it's important to note that these hormones are very slow-acting. Steroid hormones are slow-acting because they affect transcription. Now, you may be wondering, why do hormones that affect gene expression act slower? And these are things like steroids again. So first you got to consider that this is in comparison to steroids that bind to the outside of cells and set off these signal transduction pathways. And these are things like peptide hormones. So the first thing that makes these kind of hormones slower acting is the fact that they have to diffuse all the way to the nucleus to act. Another important thing to consider is the fact that changing transcription is much slow. It's much slower in nature because you have to go through transcription, then translation, and that's to create a single protein, which is much slower than the signal transduction pathway that has exponential amplification at each step of the signal transduction pathway. And I'll talk more about this in a different podcast, but just know the signal transduction pathway is more of like a rapid fire, like boom, 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 like an automatic rifle versus a pistol. All right, so back to the topic at hand. And the main thing to remember about a steroid molecule structure is it's got 4 rings. And in addition to having 4 rings, you can usually basically recognize a steroid on the MCAT by the ending of its name. So the common endings for steroid hormones are all, own, or oid. So, the classic example is testosterone. That's a lot of bodybuilders take that to get big. They use it in hormone replacement therapy for people that are transitioning, etc. So again, steroids, 2 takeaways: 4-ring structure, and number 2 is small molecule, nonpolar. All right, moving on to the next biomolecule to know. These are nucleotides. And so nucleotides are organic molecules that serve as the monomer units for forming DNA, or deoxyribonucleic acid, and also RNA, which is ribonucleic acid. And both of these are essential within all life forms on the earth. And nucleotides are, basically nucleotides are the building blocks of these nucleic acids, and they're composed of 3 subunit molecules. And so the first is a nitrogenous base, a 5-carbon sugar, and that's where the ribose or deoxyribose comes in, and at least 1 phosphate group. So the nitrogenous bases are adenine, uracil, guanine, thymine, and cytosine. And you'll definitely see these as single letter abbreviations. The purines are A's and G's, and these are 2-ring structures. And the pyrimidines are C, U, and T, and these are 1-ring structures. And the way I remember this is C, U, and T is like cute. So I remember these are small, i.e. they're cute. So small and cute. And you can remember I also remember it through the opposite way, as in, so pyrimidine's name is a lot longer than purine. So I remember the longer name corresponds to the smaller, cuter structures. And it's important to recognize these bases, you recognize the structure of these bases. And once again, recognition isn't quite as hard as, memorizing it, and so you could redraw it. So for the 2-ring structures, these are the purines again. Adenine has an amine group on it attached to one of the rings, and this is an NH2 group, amine group. On the other hand, guanine has an amide group, which is a carbon double bound to an oxygen, and then that carbon is also bound to a nitrogen. And then in addition to the amide group, it has an amine group like adenine. So again, remember that guanine has the amide group. So for the pyrimidines, if you remember, these are single-ring structures. Cytosine has an amide group and also an amine group. And then uracil and thymine are very similar structures. They both have 2 amide groups. So again, that's 2 carbons double bound to oxygens, these carbons are also bound to nitrogens. And the only difference between uracil and thymine is that thymine has a methyl group at the 5 position. So go look at all these structures, make sure you are able to recognize which is which. And make sure you also recognize the differences between the nitrogenous bases for each nucleotide. It's also important, real quickly, to talk about some of the different naming aspects of nucleotides. So you might hear the term nucleoside. So a nucleoside is just a nucleotide without a phosphate group. And then another thing you may see is monophosphate or diphosphate or triphosphate. And so these are just referring to the number of phosphate groups attached to the sugar. And also, in comparing ribose and deoxyribose, this refers to the hydroxyl group at the second position. And so deoxyribonucleotides don't have a hydroxyl group at this 2' sugar, or at the at the second carbon in the sugar, or the number 2 carbon. And then the ribonucleotides have a hydroxyl group at the number 2 carbon. So you can remember deoxy, you're just taking out the oxygen at the second carbon. All right, so to put all of this together, you might hear, or you may have heard of, adenosine triphosphate, or ATP, very important molecule. You can go through this structure. Triphosphate, it's got 3 phosphates. It is in, the base is adenosine, and so that's a 2-ring structure with a single amine group, and then it contains a ribose sugar, so it has the hydroxyl at the 2' carbon. And these 3 phosphoryl groups can be further named, so they're referred to as the alpha phosphoryl group, the beta phosphoryl group, and then the gamma phosphoryl group, which is the terminal phosphate. So again, alpha is closest to the sugar, and then going out you have beta and then gamma. So the first phosphoryl group that is lost is the gamma phosphoryl group.
And there's a very easy way to remember this. So think about the difference between an alpha and a beta. We're talking The Rock versus McLovin. Let's say this alpha and this beta are both holding onto a rope, and it's a tug of war. Who is more likely to lose? I mean, that's pretty obvious, right? It's the beta. So you can think about it like this. First phosphate group to go, obviously I just talked about, was the gamma. But then you got to remember, okay, which one's next? Which one goes next? Well, it's the weaker of the two. That's going to be the beta phosphate group. And then once that beta phosphate group has left, now you have a monophosphate. And this is the alpha. This is the strongest. This is the one that just hangs around the longest. So remember that. You can remember that order. Gamma first to be lost. Then you have the beta. Then you have the alpha.
So now that you have a basic understanding of the structure of these nucleotides, or the building blocks of DNA and RNA, let's talk about the structure of DNA. So as you probably know, Watson and Crick back in the day discovered that DNA forms a double helix. The backbone of this helix is formed from the sugar phosphate groups of the nucleotides. And then the bases kind of point inward from this backbone and hydrogen bond. So these specific groups hydrogen bond with other specific groups. C's pair with G's, A's pair with T's, and in RNA, T's and U's are switched, so A's pair with U. And it's important to remember that C's and G's hydrogen bond have 3 hydrogen bonds, while A's and T's have 2 hydrogen bonds. And so C's and G's tend to form stronger bonds. So if your DNA contains, if a certain stretch of DNA contains a greater percentage of C's and G's, it's going to be bound more tightly. It's not going to degrade as easily. And an important bond in the formation of DNA is the phosphodiester bond. And so this is what connects the nucleotides to each other. And so essentially the single phosphate group that's attached at carbon number 5 of the sugar molecule of a nucleotide attacks the 3' carbon of the adjacent nucleotide. So this 3' carbon has a hydroxyl group already attached. What happens is an oxygen that's attached to the phosphate attacks that 3' carbon, and I keep calling it 3' carbon. It's just the third carbon. So it attacks that carbon number 3, and it kicks off a water molecule, and you form a phosphodiester bond, a strong linkage. So once all these bonds, all these phosphodiester linkages are formed, you have a big contiguous DNA molecule. And so when this helix forms, you get a few interesting little things that go on. So you get a few different grooves in the DNA. So you have the major groove, which is a 22 angstrom opening in the groove. And then you have a minor groove, which is this little smaller groove in the helix that's about 12 angstroms across. And so I recommend you go look at this, but essentially it doesn't form this perfect, completely equal helice. Instead, you have it's almost bunched up in certain places, forms a minor groove and a major groove. All right, so now that I've gone over DNA and nucleotides, I want to move on and talk about the structure of carbohydrates. And so we talked a little bit about carbohydrates in terms of the ribose sugar that's in nucleotides, but I'm going to get more into carbohydrates that you eat, glucose, mannose, etc. So let's get into that. So carbohydrates can be shown in a linear form and also in a ring form. It's important to note that these structures spontaneously form rings, so they're not usually in this linear form, but the linear form is a good way to kind of visualize it. And so one thing you should memorize is the structure of glucose, the linear form, and also the ringed form. And there's two different types of sugars that you should know. So the first is an aldose. And so this is a sugar with an aldehyde group. And this means that the carbonyl carbon is the last carbon in the chain. And then you also have ketoses, which are sugars with a ketone group. And essentially, this is where your carbonyl carbon is not at the end of the chain, and therefore you have a ketone group that forms. And these ketoses form 5-membered rings, which are called furanose rings, and then the aldoses form 6-membered rings, which are called pyranose rings. And so in terms of the ring structures of sugars, you can have sugar monomers, which are called monosaccharides. You can have disaccharides, which are two sugars linked, and you can have polysaccharides, which are these really long chains of sugars. You can have oligosaccharides, which are 3 to 10 different sugars connected. They're kind of in the mid-range between polysaccharides and disaccharides. So it's important to be able to recognize and answer questions about the different stereoisomers of monosaccharides. So if you look at the anomeric carbon, which is the carbon that is connected to two oxygens, you'll notice that it can either have the hydroxyl group it's connected to pointing up or pointing down. And of course, this is relative to the rest of the molecule. What if you just took the whole thing and flipped it around, or took the whole glucose and flipped it right over, right? It'd be up or down, but it'd still be the same thing. If you keep everything the same and you just flip the glucose upside down, well, it's going to be the same exact molecule.
And so you need a reference group to look at.
And this is the CH2OH group on the last carbon. You're going to look at the direction in which this is pointing and then make your determination of what kind of stereoisomer it is. So the alpha configuration has the anomeric hydroxyl and the CH2OH group facing opposite directions or on opposite sides of the ring. And then the beta anomer or the beta stereoisomer has the hydroxyl group from the anomeric carbon and the CH2OH group on the same side of the molecule of the monosaccharide. In this case, I'm looking at glucose, so you're going to have alpha glucose and beta glucose. And it's important to understand also the numbering of the carbons. For instance, the MCAT might ask you to correctly identify glucose 6-phosphate or glucose 2-phosphate, at which point you might be like, shit, which one is carbon number 2? Well, if you haven't memorized, this will be like a piece of cake. You'll be, boom, 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 got it. So if you draw out the linear form, you can see that the anomeric carbon, or the carbon connected again to two oxygens, is carbon numero uno. And so then you can just go down the linear chain and successively number those. And another way to remember this is that the anomeric carbon, number 1, and then you can go around the ring structure. You can go around opposite the direction of the CH2OH. That's going to be the last carbon, or carbon number 6. All right, so moving on to disaccharides. One thing you need to know here is how to name the glycosidic bonds that join the two monomers together to make that disaccharide. So these are called glycosidic linkages or glycosidic bonds. And these bonds are named according to, number 1, the numbers, as I just explained, the numbers of the connected carbons and the position of the anomeric hydroxyl group of the sugar involved in the bond. So that is the either alpha or beta anomer. So again, there's 2 things to look for when naming these glycosidic linkages. Number 1, the number of each carbon, and number 2, whether these carbon are the alpha form or the beta form. So let me give you a quick example. So sucrose is a combination of glucose and fructose. And in this case, the glucose is in the alpha form. In other words, the CH2OH and the number 1 carbon, or the number 1 hydroxyl group, is on the opposite side. So those 2 are on opposite sides. And then the fructose, which is a furanose molecule, has its anomeric hydroxyl group pointed or on the same side as the CH2OH group. So this is the beta anomer of fructose. So together, you have a glucose that's in its alpha form and a fructose in its beta form. Next thing you look for is the carbon that is attached to the oxygen that is the glycosidic linkage. So in glucose, it's the number 1 carbon is attached to the oxygen of the glycosidic linkage. And in fructose, the number 2 carbon is connected to the oxygen of the glycosidic linkage. So putting it all together, the glycosidic linkage in a sucrose molecule is called a alpha 1 beta 2 glycosidic linkage. And I recommend just going over a bunch of these, practicing, looking at the molecules actually in real life. That way you can figure out, a good way to remember in your head. And then but most importantly, practice it. Do molecules that you don't know, practice naming them, and then looking up online or whatever you need to do. The last thing I want to mention real fast are the naming of 2 different polysaccharides. And so most plants store glucose in the form of starch, and these glucose monomers are joined together by alpha 1,4 linkages and alpha 1,6 linkages. So amylose is entirely unbranched chains of glucose, and these are all alpha 1,4 linkages. And then amylopectin is a more branched polysaccharide. So it consists of 1,4 polysaccharides which form a linear chain, and then it has branch points which are alpha 1,6 glycosidic linkages, and then it forms chains off of these in much more branch. And so you can remember amylopectin is a longer word, therefore that is the more branched version of starch, or which is a polysaccharide. And then lastly, I'm sure you've heard of glycogen, and this is the same as starch, but has additional alpha 1,6 linkages for branching, even more than amylopectin. And now that I've talked about glucose, I want to get into fatty acid structure. As a biomolecule, fatty acids serve many different purposes. This ranges from long-term energy storage to cell membrane makeup, and some of them even exhibit hormone-like effects. So fatty acids are stored as triglycerides, and these form body fat, and so this is the type of fat that makes up, the love handles that you may or may not have. And triglycerols are a glycerol backbone, which is a 3-carbon backbone and have 3 long fatty acid tails. So you can remember that pretty easily. Triglyceride. So 3 fatty acid tails and a glycerol backbone. The fatty acids that make up cell membranes are called phospholipids, and they can form lipid bilayers because of their amphiphilic characteristics. And all an amphiphile is a chemical compound that possesses both a hydrophilic group and a hydrophobic group. The structure of the phospholipid molecule generally consists of 2 hydrophobic fatty acid tails and a hydrophilic head consisting of a phosphate group. The 2 components are joined together by a glycerol molecule, and the phosphate group can be modified with simple organic molecules such as choline, ethanolamine, or even serine. And it's important to note that these phospholipids can either form micelle, which is a single layer, or a lipid bilayer, which is 2 layers of these phospholipids. And that's also known as a liposome. And so you've probably seen pictures of this before, but essentially you have these hydrophilic head groups pointed towards the aqueous phase, and you have the hydrophobic tails pointed inwards. And so when these phospholipid layers form, you can see these head groups pointed out, and then the interior of that membrane is composed of all these long fatty acid tails. And these long hydrocarbon tails can either be saturated or they can be unsaturated. Saturated hydrocarbons consist of these tails that have no double bonds, or in other words, they're fully hydrogenated, they're fully saturated with hydrogens. And on the other hand, unsaturated fatty acids are not fully hydrogenated, or they are not saturated with hydrogen. And in other words, they have some double bonds. And so these double bonds cause the hydrocarbon tails to become angled or kinked, as some teachers probably present it to you. They become kinked, and this affects the membrane. This affects the membrane by making it more fluid. This is because it leaves more room, essentially, between these different fatty acid molecules, or between these different phospholipid molecules. It leaves more room between them, and it makes a more fluid membrane. And you can kind of remember this with the example of butter versus oil. So oil is a pretty heavily unsaturated oil, and so you can think at room temperature that's a liquid, and then you can compare that to butter, which is a saturated fat. And you can see that at room temperature, that's a solid. So just think about that when trying to remember how the saturation of these fatty acid tails affects the membrane fluidity. Cholesterol, which has a steroid structure, and if you remember, it's got the 4 rings. It acts as a bidirectional regulator of membrane fluidity. This is because at high temperatures, it stabilizes the membrane, making it less fluid and raising its melting point. And then at low temperatures, it essentially does the opposite and prevents the phospholipids from getting clustered together and stiffening up. In other words, it makes the membrane more fluid. So how exactly does it do both of these? Well, at low temperatures, the cholesterol essentially fits right between the different phospholipid molecules and distances them or splits them apart further. And so when you're at really low temperatures, these phospholipids tend to kind of bunch up, get really tight, and crystallize. And so when you wedge a cholesterol molecule in there, it actually prevents this crystallization from happening. On the other hand, when you're at high temperatures, these phospholipid molecules are already kind of spread apart. And what happens is these cholesterol molecules are inserted into the membrane between these phospholipids. And this time, if the phospholipids are kind of spread apart, you get some van der Waals, or these non-polar kind of attractions. You get these attractions, and it kind of pulls everything in a little bit tighter. It pulls those fatty acid tails in a little bit tighter, and actually decreases the fluidity. So at high temperatures, decreases the fluidity, makes a more stable membrane. And so now that I've talked a little bit about the structure and kind of what these cholesterol molecules do, I'm going to talk a little bit about some of its implications in heart disease. And so I'm sure you've heard of this before, but LDL levels, which are low density lipoprotein levels, are a classic measure of cardiovascular disease. And LDL is referred to as the bad kind of cholesterol. And its function is to carry fats and cholesterol to different parts of the body for delivery. The LDL is the delivery vehicle. That's like that's kind of like the UPS truck. It's coming and it's delivering those packages, fats and cholesterol, to different parts of the body. On the other hand, HDL, which is high density lipoprotein, does the opposite. It picks up cholesterol and takes it to the liver for disposal. So this is the Uber driver. This is the Uber driver that comes and picks up the people and it takes them to the liver for disposal. So just like an Uber driver. With that said, though, we've kind of been getting away, or I guess the U.S. and the American recommendations have been getting away from attributing cholesterol to cardiovascular disease. In 2015, the Dietary Guide for Americans did not specify a amount of cholesterol in which to restrict your diet. This is in stark contrast to the dietary guidelines from before this time. And, you could do a whole interesting podcast on the studies that have been done on cholesterol and cardiovascular disease. You could point out some confounders that aren't really accounted for in these different papers. And, you could point out the good things about this research. This would be really interesting, but outside the scope of the MCAT. All right, so I'm going to continue, get a little deeper into the phospholipids. I'm going to talk about glycerophospholipids. So glycerophospholipids are all phospholipids, yet not all phospholipids are glycerophospholipids. So glycerophospholipids, or phosphoglycerides, are specifically those phospholipids that contain a glycerol backbone bound by ester linkages to two fatty acids and a phosphodiester linkage to a highly polar head group. And again, these are a specific subset of phospholipids. So you might be wondering, why do you need to specify the type of phospholipids that contain these glycerol backbones? Well, it's because there's another set of phospholipids, and they are called sphingolipids, and they contain a sphingosine backbone. And a sphingosine backbone can be recognized pretty quickly by looking for an amide group in the backbone. And again, an amide group is a carbonyl with attached to a nitrogen. So it's a oxygen double bound to a carbon, which is bound to a nitrogen. And other than that amide group, these sphingosines look very similar to phospholipids or even glycerophospholipids. Just remember sphingosine backbone. That is the differentiator for sphingosine. And so these sphingosine molecules have a few different functions. And so number one, they protect the cell surface against harmful environmental factors. And number two, they modulate different biological processes. This includes growth regulation, cell migration, cell adhesion, apoptosis, sustenance, and inflammatory responses. All right, so as I mentioned in the intro, the last group of fatty acids or lipids that I want to talk about are the lipids that act as hormone-acting agents. So the first group of these molecules are called eicosanoids, and these molecules are signaling molecules that are made by the oxidation of arachidonic acid. And arachidonic acid are 20 carbon long fatty acid chains. Again, that's 20 carbons long. And so basically when you see 20 carbon fatty acids on the MCAT, think signaling molecule. And so, the question then becomes, what does this eicosanoid molecule signal? Well, it's quite a very diverse array of things that it signals. And, to name a few, that's osteoclast differentiation, vascular function regulation. It also modulates blood clotting. And so there are three kind of subgroups of eicosanoids that you should know, and these are prostaglandins, thromboxans, and lastly, leukotrienes. And so I'm going to go into kind of what each of these does. So thromboxans are named for their role in clot formation, and you can think of thrombosis, which is the medical term for a blood clot inside of a blood vessel. And there's a few things to note about its structure, or I guess one thing to note about its structure, and that's its distinguishing feature is that it contains a 6-membered ether-containing ring. And so again, I recommend you go look this up and see exactly what it looks like, but just know that the thromboxans contain a 6-membered ether ring. All right, so next are prostaglandins, and prostaglandins are unsaturated carboxylic acids consisting of a 20 carbon skeleton, and that contains a 5-member ring. So the feature to recognize of a prostaglandin is the 5-membered ring. And it looks essentially like a phospholipid, but with that 5-membered ring at the head. And these function in contraction of the uterine wall. And last but not least are the group of molecules known as leukotrienes. And these are a little bit easier to recognize because these molecules are eicosanoids that contain no rings. So again, its backbone is a 20 carbon chain, but this time it does not have a ring, and it's kind of in the shape of an M. Again, look these up, kind of figure out what they look like and remember them. But importantly, it functions in the regulation of the immune system. And so you can remember this by thinking of the prefix here, which is leuco, which means white, or kind of in this case, white blood cell. And again, white blood cells correspond to the immune system. So remember, these function in regulating the immune system. All right, so brief summary of the structures. So prostaglandins, they have a 5-membered ring at the head and otherwise look like a phospholipid. Thromboxans have a 6-membered ring with an ether group in the ring. And then lastly, leukotrienes have no rings, and all of these are 20 carbons long. And quick note, so if you remember, it's called a 20 carbon molecule is called, or in this case, it's called arachidonic acid. And so it kind of looks like the word arachnid. So I try to imagine a spider with 20 legs, and that's how I remember these different lipid hormone signaling molecules. All right, so now that I talked about fatty acids, the last group I want to talk about is the heme group. And you're likely to see some kind of question on the heme group. And likely you'll be asked about it in terms of hemoglobin. So what exactly does this heme group do? And what is this heme group's structure? So a heme group is a prosthetic group made of a warfarin ring that is conjugated to an iron atom. And obviously this is kind of a very complex, convoluted definition, so let me break that down. So first, it's a prosthetic group, which means it is a tightly bound, non-polypeptide component of a protein that is required for that protein's function. You can think of this like coffee to a student. And you're a student, how well do you study for the MCAT without coffee? I know for me, the answer would be I would be non-functional and I wouldn't be able to study without coffee. So a warfarin is a very large aromatic ring structure that contains nitrogens. And so again, this is something that I recommend you look up. It's basically just this massive ring that's aromatic. And so it often supports these metal groups in the middle, or metal complexes that are in the middle of this ring. And in the case of heme, this is an iron atom. So to just throw all this together for you real fast. So a heme group is a prosthetic group from which proteins derive their function. and it is made of this big aromatic ring called the warfarin group, and in the middle of this warfarin is a conjugated iron atom, and this iron atom functions to bind oxygen. And again, this is in the case of hemoglobin. So obviously hemoglobin binds oxygen. Well, now you know that the iron atom is really what gets this done. So I want to touch on some kind of other functions of heme groups. Obviously, you might see hemoglobin, but what are the functions of these heme groups in other proteins like? Well, in other proteins, the heme group essentially can serve as a source or a sink of electrons during electron transfer reactions, redox chemistry. Okay, cool. So now that I've talked a little bit about heme groups, I'm going to start the last little subject here, which is a brief talk about how the structure of these molecules is related to their entry into cells. So first I'm going to talk about carbohydrates, then talk about amino acids, nucleotides, and then small nonpolar molecules and how these all get into the cell. So carbohydrates are too large to diffuse across the membrane, and therefore they must be transported via membrane transport protein, and these are called GLUT2 and GLUT4 membrane proteins. Amino acids, on the other hand, are quite a bit smaller than carbohydrates. However, they're very polar, so they still need to be transported across the membrane. Nucleotides are also pretty small molecules. However, they are also too very polar and therefore require a transport protein. And then lastly, we have these group of molecules that are small and nonpolar, and these diffuse straight through the membrane. And so these include your steroids, your lipids, and also alcohol. And so to kind of sum those up in two easy rules to remember, number one, only relatively small nonpolar materials can move through the lipid bilayer or the cell membrane. And remember that these long kind of tails which form the interior of the lipid or the interior of the lipid bilayer are nonpolar themselves. And rule number two is water-soluble materials like glucose and amino acids and electrolytes need some assistance to cross the membrane because they are repelled by those long hydrophobic tails of the phospholipid bilayer. And I know I said there's only going to be two rules, but kind of a common sense third rule is that huge molecules are going to need some help getting across the membrane. So these huge, say you have a huge fatty acid chain, sometimes those big chains will need help getting across the membrane. So that's something to keep in mind is the relative size of the molecule. All right, so that is it for lesson number 5 on biomolecule structure. The next lesson number 6, is on enzymes. So look forward to that one. Thank you for listening. Hope you come back again for the next one. All right, so now time for the MCAT Advice of the Day segment. All right, so this advice might sound kind of silly, but this is something that worked for me. So, the night before you take the test, it might be pretty hard to fall asleep. I know I was pretty anxious, and the thing is, though, I didn't have trouble falling asleep because what I did is I went out that day. I totally took a break from even looking at anything about the MCAT. I tried not to think about it. I didn't want to do any studying whatsoever. That was just kind of a break day. But what I did do is I did basically the hardest workout of my life. I did a shit ton of cardio, and then I lifted weights, made sure I was exhausted, and this was basically all in order to be able to fall asleep really easily the night before. I didn't want to take like NyQuil or some sleep aid because I was worried about waking up and feeling kind of drowsy. And that did the trick. I slept like a little baby, and I felt pretty good for the test the next day. If you've ever taken a test while you're tired, it sucks and it's harder. So that was my strategy. Work out super hard. That way I was really tired for that night and was able to sleep well for the test. I get that might be kind of specific for my case, but hopefully
If you have trouble sleeping and get anxious about tests and stuff, this might be one strategy you might think of.
Each episode of MCAT Basics is brought to you by Med School Coach. To access Med School Coach services, including MCAT tutoring and medical school admissions advising, visit our website at medschoolcoach.com. Good luck as you prepare for the MCAT, and we hope you tune in again next time.
Come on, Murph. I'm trying to podcast over here, dude.